Good evening, everyone. I invite Deepali ma'am to conduct this session.
Okay, fine, sir. So ready? Everyone ready?
Yes, ma'am.
Okay, fine. Then start the class. So I am taking the yesterday session again because yesterday we are having technical glitches. Okay. So let's take the yesterday topic and start from the beginning. Is this visible?
Yes, ma'am, it's visible now.
Okay, fine. Okay, so let's start. Unit 2, structural organization of animals. So before we start this topic, we need to understand the classification of living organism. So this is the basic classification of living organism. Living organism basically mainly divided into two types, that is prokaryotes and eukaryotes. Prokaryotes are only unicellular, and you will be studying in detail about the prokaryotes in Monera kingdom. Okay, eukaryotes are unicellular or multicellular. They are of two types. Unicellular protista, multicellular fungi, plantae, and animalia. Fungi and plantae will come under the botany, and zoology. In zoology, animalia we will be studying animalia. So let's start the topic. So have you heard about the hierarchy? You must be knowing the hierarchy of organization of our body. Cell, then from cell, after cell, tissue, after tissue system, then organs, then organ system, and then organism. Okay, so this is the grades of organization and body plan. So animals show different shapes and sizes. They are of different shapes and different sizes, but but their organization is either cellular, tissue, organ, or organ system. Means grade of organization does not depend on the size and shape. Okay, so they can be cellular grade of organization. They must, or they are having tissue kind of organ type or organ system kind like us. Okay, cellular grade of organization is done only in unicellular or acellular organism. Okay, like amoeba, paramecium, etc. Animal cells are diverse in structure. What animal cells are diverse? Means they can perform various, their structures are so diverse, they can perform many functions. But what they are lacking? Cell wall, and quite flexible. So the main difference between the plant cell and animal cells is a cell wall, presence of cell wall. Plant cells are having cell wall and animal cells are lacking cell wall. So here comes the few examples of unicellular organisms. Here you can see paramecium. Parameciums are also known as sleeper animal fuel because their shape is like sleeper. Okay, they are having two nucleus, macronucleus and micronucleus. They are ciliated, and another is amoeba. Amoeba, you know, these are the pseudopodias. Pseudo means false, legs. And this is euglena. Euglena is a connecting link between plant and animal because it contains chloroplast. Chlorophyll is present in euglena, so perform photosynthesis. So these are few examples of unicellular grade of and organization. Okay, they are performing all the physiological functions like excretion, respiration, digestion, and many etc. Many functions they are performing only via help of single cell. So this is cellular grade of organization. Now come metazoans. So the next higher level of body organization, means multicellular organism is observed in multicellular organisms, which are called metazoans. So metazoans are further divided into two types. Okay, one is parazoa and one is eumetazoa, into two subkingdoms. So what is parazoa? In parazoa, cells are loosely aggregated. They are not compactly arranged. They are loosely aggregated and do not form tissue or organs. Example, sponges, phylum, porifera. Or you can say euspongia also. metazoa. Okay, in metazoa, cells are organized in tissue grade of organization or organ grade or organ system type of structural and functional units. Means they are more advanced, they are more complex organism. Okay, body plan either sac-like in cnidarians like hydra or tube-like in rest of the eumetazoans. Body plan means metazoa is further divided into two types. If their body is like having a sac-like structure or a tube-like, okay, like in parazoa, you can see this. This is an example of parazoa. These are unicellular. Okay, only one ostia or pore. This is a single cellular rate. Ostia is a pore. Water enters from the ostia and comes out from the osculum. So with the help of water current, they perform respiration, excretion, and many other things. Okay, and they are sessile. Sessile means they are attached to the bottom of the ocean. They cannot move. Okay. Next is blind sac eumetazoans that we are talking about. Metazoans are two types on the basis of either they are sac-like, having a sac-like cavity, sac-like body structure, or a tube-like body structure. So sac-like is a hydra, cnidarians. This is an example of hydra. You can see here the sac-like structure. Okay. So these are having one more thing. These are having, this is oral end and this is aboral end. These are also sessile. They are also attached to the oceans. And with the help of these tentacles, it attacks its prey and then takes the prey inside this or through the active oral entry. And then some more hydrolytic enzymes are present in this sac. So digestion is performed. And what is the mode of reproduction? As you can say, the bud is arising. So asexual mode of reproduction. This bud will develop, grow, and then detach from the main body and attach to the ocean and develops into a new hydra. So this is the mode of asexual reproduction. Now comes to symmetry. Symmetry, as you have seen in parazoa in sponge. Can you see this? Can you see any symmetry in this? No, these are asymmetrical. What is asymmetrical? We cannot divide this organism.
Which cannot be divided equally.
We cannot divide the organisms into two equal halves. From whatever end you are cutting, from whatever axis you are following, you cannot divide the organism into equal halves. Okay, this is the asymmetrical. So means parazoa, phylum porifera, they are asymmetrical. You must be getting these MCQ questions in your exam that tell us the example, or you might be getting few examples of asymmetry. If you are getting sponges or porifera, then just tick it. These are asymmetrical and unicellular also. Now symmetry. Parazoan animals lack any definite symmetry, means they are asymmetrical. Example, sponges. Now eumetazoans can be further grouped into two categories based on the symmetry. Either they can be radiata, radial symmetry, or bilateral, bilateral symmetry. Okay. Now what is radial symmetry? When you are cutting any object from a plane that is passing through the center, and you can divide that organism to equal two halves with mirror imaging. This is radial symmetry. Okay. When any plane passing through the central axis of the body divides the organism into halves that are approximately mirror images. Okay. See an example. If you are having an orange or a lemon, if you are cutting the lemon from the center, you can divide the lemon into two equal halves that are the mirror images of each other. Okay. It is called radial symmetry, and animals are called radiata. Examples, cnidarians, sea anemone, corals, jellyfish, and ctenophores. These are comb jellies. So what is this? From up to peripera, protozoa and porifera phylum, they are asymmetrical phylums. Okay. From cnidarians, from the third phylum, you get the radial symmetry. Okay. Sea anemone, corals, jellyfish, and ctenophores. So cnidarians and ctenophore phylum is having radiata symmetry, radially symmetrical. These animals have oral and aboral end. I have shown you in the hydra. You can see here, this is oral end and this is aboral end. Okay. They are not having left and right part. Okay. They are not having left and right part. Bilateral symmetrical organisms are having left and right part. Now comes, here is a diagram. What is radial symmetry? This is an drawing of an actinomorphic cloud. If you are dividing from here or from here, it doesn't matter. From where, whatever point you are dividing, but just you need to pass through the center. This is radial symmetry. Okay, what is spherical symmetry? This is spherical symmetry. Coccus, bacteria, streptococcus, all those bacteria are radial. Cocci means circular shaped bacteria. You must be in further exercise, in further chapters, you must be reading about the structural organization of bacteria too. Okay, then I will explain you about the cocci, streptococcus, and all these spiro, comma-shaped bacteria. I will be explaining you everything in detail in that chapter. Okay, now comes to bilateral symmetry. In the remaining eumetazons, means after Hydra and Ctenophore phylum, comes all the other phylums from invertebrates to chordates. All are bilaterally symmetrical. What is bilateral symmetry? In the remaining eumetazons, the body can be divided into two identical left and right halves in only one plane. If you are cutting your body from here to the end, then you will divide your body into two halves. Okay, this is bilateral symmetry, and that two halves is one is left part and other is right part. This kind of symmetry is called bilateral symmetry, and animals are called bilateria. These animals have front or anterior end, like we are having this is front end and this front end, and we are having also posterior end. Okay, and dorsal or ventral part. What is dorsal part? This part of our body is called ventral part, and the back side of the part our body is called dorsal part. This you can see, you can understand this in fish mode. Have you seen the diagram of fish? Okay, I'll explain you. Have you seen the diagram of fish? The anterior part of the fish is called anterior part, and posterior part is pelvic fin. Okay, pelvic fin is the posterior part, and when you cut the fish through the axis, you can see the upper part is dorsal part and the lower part of the fish is ventral part. Okay, so these are bilaterally symmetrical animals. They are having this front or anterior part, anterior, posterior, and dorsal or ventral part, like fishes. Now comes symmetry diagrams. Asymmetrical organism. You have seen, I have explained you in the sponges also asymmetry. Now comes amoeba. Amoeba is completely asymmetrical animals. Why? Because there is no definite shape. Amoeba changes the shape because amoeba needs pseudopodia for the movement. So amoeba extends the pseudopodia and changes its shape. So asymmetrical organism. Any line, and what is radially symmetrical? Starfish. This is an example of starfish. Any line passing through the center will divide the body into two similar halves. Means you need to pass through the center, okay, to divide the organism, like starfish. These are radially symmetrical. The body can be divided into two similar halves only through one plane, that is along the middle axis. This is bilateral symmetry. You can see the diagram of human being. If you are dividing the human being from head to toe, you can get the two human beings, two parts of the human being, the left part and right part. This is the example of bilateral symmetry. Now comes the germ layer division. How we divide the organisms on the basis of germ layer? Diploblastic and triploblastic. Di means two, tri means three. Blastic means when embryonic development takes place inside the womb of a mother, means when embryo is developing. So embryo is a 8-celled structure. Okay. So when embryo is dividing its cells further, so that small cells are called blastomeres. It forms a blastopore. Means it forms the 3 germ layers. Okay. Or 2 germ layers. 2 germ layers. What are 3? Endoderm, mesoderm, and ectoderm. Okay. If they are diploblastic, then only 2. Mesoderm is lacking. Mesoderm is missing in triploblastic. And triploblastic, endoderm, mesoderm, ectoderm. Or you can say ectoderm, mesoderm, or endoderm. And radiate animals are arranged in 2 fundamental layers. Means radial symmetry organisms are having diploblastic germ layer organization. Diploblastic means they are missing mesoderm. And radiate animals are arranged in 2 fundamental layers, an external ectoderm and internal endoderm. So there is a layer between ectoderm and endoderm, that is mesoglia. Mesoglia is a non-living layer. Why we are saying non-living layer? Because the cells of mesoglia are not dividing. Okay. Such animals are therefore called diploblastic. Example: corals, hydra, sea animal. These are the examples of diploblastic organisms. This is the diagram of a germ layer. With the help of this diagram, it will be easy for you to understand this. Okay. You can see the blue part. This blue part is ectoderm. This light pink part is mesoglia. This is a non-living layer. And the bright pink part is endoderm. And what is this? This is a gut. Okay. And this is triploblast. In triploblast, green color mesoderm is present. So this is having 3 layers: endo, meso. Okay. This is another example. You can see this green, pink part. This is mesoglia, non-living germ layer, ectoderm and endoderm. And in triploblastic, you can see the orange layer with slight pink out boundary. This is a mesoderm. Okay. This is germ layer. One more diagram. Blue part is mesoglia. Yellow part is mesoderm. So this is a difference between diploblastic and diploblastic. That diploblastic are having mesoderm as a third layer, and diploblastic is lacking in mesoderm. In spite of mesoderm, they are having a non-living layer, mesoglia. Okay. Now, what is triploblastic animals? Here is a definition. In bilateria, a third germ layer, mesoderm, is present between the ectoderm and endoderm. Okay. A third germ layer, mesoderm, is present between ectoderm and endoderm. Means endoderm and ectoderms are separated by a third layer, that is mesoderm. Hence, they are called triploblastic animals. Tri means 3. Plastic means they all are formed from blastomeres. Okay. So that's why they are called triploblastic. In these animals, embryonic blastopore forms mouth in protostomia and anus in deuterostomia. This you won't be understanding what is protostomia and deuterostomia. I will be explaining you. Now, what is means triploblastic animals are further divided into 2 categories. One is protostomia, another is deuterostomia. Why? What is the basis of division? See, when an embryo is developing into an organism, so it depends if the anus is developed first, then the animals are deuterostomia. And if the mouth is developed first, animals are protostomia. Okay? Here's a bilateral subdivision. Protostomia and deuterostomia. Blastopore gives rise to the mouth. Blastopore gives rise to the anus. Cleavage is spiral and determinate. What is cleavage? When I have told you 8-cell structure of an embryo, when embryo is dividing, then cleavage is spiral in protostomia. Spiral means cleavage is taking like this. And cleavage is radial. Radial means cutting from any axis to the center part. Okay. And determinate, what is determinate? Means each cell of the protostomia are determinate that you have to make this part. You have to make vacuole. You have to make something other new nucleus. You have to make some other organelle. Cleavage is radial and indeterminate. In human beings or in higher organisms, embryonic stages are indeterminate. Okay. Why indeterminate? Because each cell of embryo is a stem cell. Dedifferentiation of cell takes place later on. Our cell does not know that whether you have to be a kidney cell, whether you have to be a liver cell or lung cell. That is why embryo stem cells are preserved when a baby is taking birth. If you are preserving if someone preserves the stem cells, you must be knowing that stem cell preservations are on in the country, in the world. Like I have done. When I have to preserve the stem cells of my body, when I was giving birth to my first son, the stem cell persons came and they took the placenta of mine. Why? Because placenta was having the cells of embryo. Okay, all those are stem cells. In stem cells, you can develop in lab with the help of stem cells, you can develop an organ. Right now, this advancement is not so easy, but I have preserved because I can predict the future of the biology. That in coming years, many scientists will be able to make a gland, make a organ with the help of those stem cells. If my son will be needing, if my his sibling will be needing the stem cell, he can use that stem cell. Okay, these are stem cells, means these are indeterminate. Every cell of embryo is indeterminate. Dedifferentiation takes place. Differentiation means whether this is a cell. Now this cell has to be a liver cell. That differentiation takes place at the later stages. So at the embryonic development, every cell is indeterminate of higher organisms like us, like chordates. Enterocoelic, okay, cytosolic coelom. Cytosolic coelom we will be studying in detail in this chapter. Cytosolic coelom is a cavity. Okay, this is a cavity inside our body. It is very important because our visceral organs lie in this cavity. What are visceral organs? Your whole digestive organs, your heart, your kidney, your liver, your lungs, all are visceral organs. Okay, so these are visceral organs. And enterocoelic coelom means a complete developed coelom is enterocoelic coelom. And cytosolic coelom is slightly lower grade coelom. It is a true coelom, but it is not more developed like enterocoelic coelom. Now, what is segmentation? Here comes the segmentation. In some bilateria, the body is of many segments. Some animals are segmented, like earthworms. Have you seen the earthworms? Earthworms are having segmented bodies. Have you seen the nerees? Centipede, millipede. Lots of millipedes, centipedes are coming out of the soil because of the heavy rains across the country. Yesterday, I have seen so many nerees in my garden area. Okay, these are annelids. Nerees, leeches, earthworm, they all are annelida. Phylum Annelida. You will be studying all these in the phylum Annelida. Earthworm is very important. If any soil is having earthworm, that soil is very fertile. Why? Because earthworms are farmers, good friends of farmers. They fix the nitrogen of the soil. So this is slightly away from the topic. Let's focus on the segmentation. In some bilateria, the body is of many segments, which shows serial repetition of parts. Metamerism. This is called metamerism. If you see the earthworm, you can see segments, body is segmented. If you see nerees, centipede, millipede, so many legs, they all are segmented. Leeches, they are also having segmentation. This kind of segmentation is called metameric segmentation, and the phenomenon is called as metamerism. Okay, this is the quality or specialization. Yeah, you can say this is the important topic of annelidas. Annelidas are segmented. They show metamerism. This is an example of earthworm. You can see these segments. This is called metamerism. Okay. Now comes to the coelom. Now, what is coelom? Coelom is very important because when you will be studying the classification of all the phylum, important classes and everything, you will be studying these words: diploblastic, bilateral symmetry, radial symmetry. So you need to understand these topics, that what is all this. Okay, so coelom. The building plan of bilateria is achieved from 3 primary germ layers. This I have explained that 3 primary germ layers are our skin, or our layers are 3: ectoderm, mesoderm, and endoderm. So the space between body wall, what is body wall? This is body wall. Space between body wall and alimentary canal and body wall, space between alimentary canal and body wall remains lined by mesoderm. It is lined by mesoderm. Okay, this is your ectoderm, inside endoderm, and the space between body wall and mesoderm remains lined by mesoderm and is called coelom. Means if coelom is lined by mesoderm, then it is a coelomate. Okay. Now understand this with further slides. Now coelomates are coelom. On the basis of coelom, organisms are further divided: acoelomates, pseudocoelomates, and coelomates. Acoelomates means, it as the name suggests, A means absence. Acoelomates. Coelom is absence. Pseudo means false. Coelom is present, but false coelom means true coelom is not present. What is coelomates? Coelomates means they are having developed coelom. Okay. Now this is just a general diagram of coelom. You can see blue color ectoderm. The inner color is mesoderm. This red color is mesoderm. This yellow color is endoderm. And this C type structure is coelom lined by the mesoderm. Okay, so coelom is basically formed by a mesoderm. Okay, it is a cavity that is lined by the mesoderm. Okay, this is coelom, and this is digestive cavity. Digestive cavity, gut, or you can say gut. Okay. Now, what are acoelomates? As name suggests, they lack coelom. Due to solid nature of mesoderm. What is solid? Means solid nature of mesoderm. This depicts that coelom must be fluid. Okay, it is fluid-like structure. So, due to the solid nature of mesoderm, just go on the word. It will be easy for you to memorize the things. Whenever you will be revising, just read the words, follow the words. You will never forget. If you go through the words, you will never forget anything in the exam. Trust me. They lack coelom due to solid nature of mesoderm. What is solid? Means for coelom, for to be a coelom, it should be fluid. This clears from this line. Solid means acoelomates are having solid mesoderm. The space between endoderm and ectoderm is filled with mesenchyme and muscle fiber. Why they are solid? Because muscle fibers and mesenchymes are forming the mesoderm. So this is solid. Okay. Acoelomate animals. Now group includes acoelomates. Porifera, Coelenterata. Coelenterata means hydra and everything. Ctenophora, flatworms. What are flatworms? Do anyone know flatworms? Aschelmans. Okay. The insects, the parasite that comes inside the stomach, and you will develop white patches around your body, and you go to the doctor, and doctor say, oh, you are having worms in your stomach. That are flatworms. Comes from the infected food, from unhygienic spaces. If you are touching unhygienic spaces and eating with the same hand, you will get this infection. Okay, flatworms, Aschelmans, or you can say Ascaris lumbricoides. This is the common. Mainly kids below 5 years are more affected because their immune systems are less developed. Okay, Rhinocella, Nimerstina have no body cavity at all. They are not having any body cavity. Semi-solid mesodermal tissues between the gut and body wall hold their organs in place. Now, this also suggests, read the line, semi-solid mesodermal tissues between the gut, means gut and body wall hold their organs, means coelom, main function of coelom is to hold the body organ. Okay, so between the gut and body. Now, so how their organs will be kept? Their organs will be kept by the semi-solid mesodermal tissue intact. Okay, so they are lacking coelom. So the function of coelom will be done by the semi-solid mesodermal tissue. What is the function? As the line suggests, function of the coelom is to keep the organs body and hold their organs in place. To hold the organs in the place. If we are not having coelom, just example, if we are lacking coelom, then our lungs will be moving here, kidney will be moving anywhere, liver will be moving anywhere. So coelom's main function is to keep the internal organs intact. As organs are attached to each other. Okay, my organs or everyone's organs, lungs are attached to the pericardial cavity. And pericardial cavity is also carrying the heart. Okay, and diaphragm beneath the lungs, attaching the stomach, and then stomach is attached to the small intestine, then large intestine, and to the anus. So all the organs are attached. They are basically attached. This role is played by coelom to keep our organs at the right place. Coelom also allows the free movement or muscle elongation of these organs, but to keep them intact, attached to each other, done by coelom. So to be a developed organism, coelom is very important. Okay. Now this is an example of coelom in acoelomate. Means acoelomate platform. This is an example of Aschelmans. As if you will cut a section of Ascaris and you will stain and see under the microscope, you will be seeing like this. They are having ectoderm, mesoderm, and endoderm. You can see their ectoderm is quite solid. Okay, it is like semi-solid, made up of mesenchyme and muscle fiber. And this is their digestive cavity. Ascaris lumbricoids is the best example to understand the acidosis. Pseudocoelomates. What are pseudocoelomates? Pseudo means false. Pseudo means false. Means false coelom is present. So what are pseudocoelomates? Pseudocoelomate animals which have a fluid-filled body cavity. So they are having fluid. Okay? They are not semi-solid or solid. They are having fluid-filled body cavity. Tissues derived from mesoderm partly line the fluid-filled body cavity of these animals. Means they are having fluid-filled cavity but partly and lined by the mesoderm. Means the whole mesoderm is not forming a whole coelom. Only part. This you can understand with the help of example more. Just go through these lines. Thus, although organs are held in place loosely, pseudocoelomates organs are placed loosely. They are not as well organized as in coelomates. Okay. All pseudocoelomates are protostomes. Protostomes means their mouth develops first during the embryonic development. However, not all protostomes are pseudocoelomates. All pseudocoelomates are protostomes. This is clear. But all protostomes are not pseudocoelomates. This line will help you to in the MCQ. Okay? Yours MCQ comes like this. All pseudocoelomates are protostomes. However, not all protostomes are pseudocoelomates. Both of the above are right. None of the above was right. Okay? So this will be easy for you to answer that question. An example of pseudocoelomate is the roundworm. What is roundworm? Taenia solium. We are the secondary host of Taenia solium. Primary host is pig. Whosoever is eating the pig meat, pork, they usually get infected with roundworm. Okay? A pseudocoelomate or blastocoelomate in any invertebrate animal with a 3-layered body and a pseudocoelomate. Okay? Invertebrate animal means they are not having vertebra. Means we are talking about the non-chordates. This is the example of diagram of pseudocoelom. You can see this circular oval shaped structure. This is pseudocoelom. This is pseudocoelom. These are mesoderm. Okay, ecto, endo, gut, meso, and this cavity is pseudocoelom. Okay, clear? Now comes to the true coelom, that is coelomates. Coelomata, yeah, eucoelomata, have a body cavity called a coelom with a complete lining called peritoneum derived from mesoderm. Okay, with a complete lining called peritoneum derived from mesoderm. Matlab mesoderm is forming a lining that is known as peritoneum. Okay, so coelom is completely lined with a membrane with a lining that is known as peritoneum derived from mesoderm. Means origin of peritoneum is mesodermal cells. Okay. The complete mesodermal lining allows organs to be attached to each other so that they can be suspended in a particular order while still being able to move freely within the cavity. There's a cavity, alimentary canal, and there's a cavity surrounding the alimentary canal. So, like if you are eating too much, your stomach will never burst. Okay, if you have eaten too much, because coelom is allowing your stomach to enlarge up to some extent. If you will eat up to that, above that extent, then you will vomit. Okay, but your stomach will not burst. So this is the role, this is the function of coelom. Yeah, this is the significance of coelom. Most bilateral animals, including all the vertebrates, are coelomates. Means vertebrates means those having vertebra means chordata. Okay, chordates. Coelom developed in triploblast. This is clear because diploblastic germ layer origin, they are having non-living layer. They are not having mesoderm. So mesoderm is not missing. Then how they will develop coelom? Because coelom is developed with the mesodermal cells. Coelom developed in triploblast, but was subsequently lost in several lineages. What is several lineages? Lineages means as the animals are evolving, due to evolution, some lineages means some organisms are lacking this, are lost in coelom. Okay, like pseudocoelomates. Pseudocoelomates are were having coeloms, but due to mutation, due to evolution changes, and their coelom was missing. This is the quite deep part, so I don't want to explain you it's so deeper because this is beyond your syllabus. Okay, now coelomates are further divided into three parts: cytocoelom, hemocoelom, and enterocoelom. What is cytocoelom? Let's move. Cytocoelom develops from split in mesoderm. When mesoderm splits and found in annelids, arthropods, and molluscs. Now development of cytoplasm. Here you can see blastopore. This blastopore forms archentron. What is archentron? Archentron is a gut. This is embryonic development diagram. Okay. So here you can see mesoderm. This mesodermal cells, they are generating this mesoderm is splitting. Okay. And this mesoderm is in turn forming this coelom. So this is cytoplasm. This is mouth and this is anus. This will develop into mouth and this will develop into mouth and anus. Okay. This is a embryonic development diagram, how cytocoeloms develop. So you can see this is not a true coelom. Matlab, the whole circle is not made. This is cytoplasm. Okay. Examples: annelida, arthropoda, and mollusks. Any two examples of annelids are, there are so many examples: nidis, centipedes, millipedes, earthworm, leeches. Arthropods, cockroaches, insecta, all insects are arthropods. Cockroach, spiders. The arthropod is the largest phylum and having a great evolution. Great evolution by arthropod and the largest phylum because it covers insects, ants, everything. There are so many cockroaches, so many butterflies. So I will not say so many, but insects are so many. Okay. Enterocoelomates. What are enterocoelomates? A true coelom reduced and cavity filled with circulatory fluid. Okay. So why I am using circulatory fluid? Because enterocoelomates are arthropods and mollusks. Arthropods and mollusks means invertebrates. Okay. Invertebrates are not having blood as their circulatory system. They are having hemolymph. What is hemolymph? They are not having hemoglobin. They are having hemolymph. So, in spite of using hemolymph, we can say the circulatory fluid of true enterocoelomates. What are enterocoelomates? Arthropoda and mollusca. And now you will say that you are saying cytocoelomates are also arthropods. So, arthropod is a big phylum. It's a very big phylum. Some arthropods are having cytocoelomates and some are enterocoelomates. Okay. And mollusks. What are mollusks? Pearl oyster, sepia, octopus. Okay. All these are mollusks. Mollusca phylum. 8th phylum. Hemocoelomates. Now here comes our story. We are hemocoelomates. The most developed coelom. It develops from wall of the embryonic gut. Embryonic gut. I have shown you the diagram. And archentron. It develops from the wall of embryonic gut, found from echinodermata, means 9th phylum to chordata. Blood or circulatory blood flow in this cavity. Okay. Why I am using circulatory fluid? Because echinodermata is also not having red blood. They are also having hemolymph. Okay. Have you ever seen? No, na? Echinodermata means, I give you an example. Echinodermata starfish, sea urchins. Okay. Blood or circulatory blood flow in this cavity means they are hemo. Hemo means blood. Coelom means, you know, coelomate. So name suggests hemocoelomates. Always go on the name and then it will be easy for you to answer the correct MCQ. Okay. Hemocoelomates. Now comes the function of coelom. Why we are studying the coelom? Why I am emphasizing coelom too much? Okay. Why it is very important? A coelom can absorb shock or provide a hydrostatic skeleton. Okay, what is hydrostatic skeleton? Hydro means water. Hydro means water. Static means at one place, not flowing. Okay, so hydrostatic skeleton. Skeletons here depicts like something strong that is protecting us absorbing shock. Sometimes, okay, if I fall from the chair, okay, my coelom will absorb that shock. Okay, that is hydrostatic skeleton. Coelom acts as a hydrostatic skeleton. It can also support an immune system. How immune system is supported? they are having some cells. These are called coelomites. Okay, coelomocytes. They may either be attached to the wall of the coelom or may float about in freely because coelom is a fluid-like structure. It is having a blood. Okay, so whenever blood is there, immunity will definitely come. Why? Because WBCs and RBCs, they are having immune cells. WBCs are having neutrophils, NK cells, and everything that we will be discussing in detail in immunology or in the in the circulatory system, body fluids, in the chapter of body fluids. Okay, the coelom allows muscles to grow independently of the body. How? Because your large, sometimes some patients might be having some issue. Their small intestine enlarged and they are having digestion issue. So why they can enlarge? Because coelom is having that margin that muscles can grow, muscles of organs can make them enlarge. Your stomach, if you will eat too much, this example I am giving you again because it is easier to understand. If you will eat too much, your stomach will enlarge. Okay, this enlargement is provided by the coelom. Okay, this feature can be seen in digestive tract like stomach, small intestine, large intestine. Okay, significance of coelom. It plays an important role in the progressive development of complexity of structure. Means progressive development means, why progressive development? Our organs. Okay, this is progressive development. And complexity of structure means our system, our human being system is more complex. Okay, so complexity of structure comes with the development of coelom. It protects internal organs from external shocks, like our heart is protected. Okay, our intestinal organs are protected. It provides flexibility to what? Okay, flexibility means if your organ wants to enlarge, if your organ is enlarging, you like you are respiring and your lungs enlarge. When you inhale, your lungs enlarge, and when you exhale, your lungs contract. So this inhale, exhale, or lungs are contracting and relaxing. This is provided by the coelom. Okay, provides flexibility to the body. Provides, like if you are bending, if you are doing yoga, now someone can touch the knee to the head, or someone can bend and touch the uh, floor with the head. So this is the flexibility. Okay, our organs are lined by the coelom. It provides formation of movement and opportunity or enlargement of viscera. Viscera means visceral organs. Enlargement, heart enlarge. When heart pumps out the blood, okay, it enlarge. When it gets the blood, it enlarge. When it pumps out the blood, it contracts. That contraction of heart is because of visceral organ are protected with the help of coelom. It works as hydraulic skeleton. Hydraulic means as watery skeleton and circulatory system. Okay, it works as circulatory system because blood is flowing in the hemocoelomates. Okay, that's all for now. Anything, any question from anyone?
No, ma'am.
No questions? Is anyone having any question?
No, ma'am.
Is this clear to you?
Yes.
Okay, so in the next class, I will be asking questions from you because you all said that it is clear to you. So be prepared for the
Yes,
next
okay.
asking random questions from this topic. Okay? Clear?
Yes, ma'am. Any issue?
No. Okay. You have lost your choice.
Yes, Deepali, ma'am.
as far as how do you give a condition for the circle?
Is anyone having any question? Any question from anyone?
Ma'am,
Oh, yes, tell me.
does sponges have asexual reproduction?
Sponges have asexual reproduction. They are
Which
also,
type of which type, ma'am?
what type? Can you repeat your sentence?
Which type of asexual reproduction is present in sponges?
Budding. They
Okay,
form
ma'am.
buds.
Thank you.
Okay?
Okay,
Buds.
ma'am.
They form buds and buds then develop and attach to the ocean. More questions? Go through. Now, may I ask some questions? May I ask some questions? So, give me one example of deuterostomia. Okay, no one. Okay, go through this topic and I will ask you the questions in the next class. Okay?
Ma'am, excuse me, ma'am.
Yes, tell me, tell me.
Ma'am, what you are showing on screen, can you send that PDF?
Beta, can you repeat your question? Hello?
Ma'am, what you are showing on screen, can you send it that photos as
I will share everything. These are the these are the notes. I will share everything once you
PDF?
complete the whole unit. I will share whole PDF. Don't worry about that.
Okay, ma'am.
Okay? I will share everything.
Okay.
Even important questions, even more stuff than this. Okay? Any questions from anyone? Just go through this topic. This is clearly given in your NCRT. I have taught you more than your NCRT because you all are NEET aspirant. Okay? So you need more preparations.
Yes, ma'am. That is the reason I'm asking notes.
And I will be preparing some MCQs from this topic. And I will be
Madam,
putting some MCQs tomorrow. Okay?
Madam, we
And you
don't
all
have
have to do that.
NCRT books.
Okay? Can you
We don't
repeat
have
it?
NCRT books.
You are not having NCRT books. That's okay. If you are not having, just go through the topics, go through the notes that I'm providing because I am covering NCRT as well as I'm covering beyond NCRT. Okay? Because whatever you will be needing to qualify for the NEET that I'm giving you. Okay? What are the basically what are the questions from this are either they ask about the organism and they ask are this about their coeloms or they ask about their symmetry or they ask about their germ layers. So you need to understand that which phylums are having germ layers division, phylum wise germ layer division, symmetry division. You need to understand that. Okay. I will provide you this PPT once our chapter, our topic is completed.
Yes, ma'am. These notes are in very deep topic. That's why I'm asking.
Okay, fine. Okay, fine. So anything, any question, or should I end the class? Take your time, ask me the question, whatever is in your mind, ask me. Apart from the chapter, apart from the topic.
Ma'am, what is the actual difference between algae and blue-green algae?
Algae and blue-green algae. Blue-green algae
Yes.
are having chlorophyll, and algae are having different kind of pigments. So algae is a very vast. Blue-green algae is a like it's you can consider it under bacteria also or into algae also. Okay, it's a first organism of evolution. Okay, do you know the age of Earth? 4.5 billion years ago, Earth evolved. So the primitive organism that we saw on the Earth, living organism, is blue-green algae. Okay, BGA. You can consider it as a cyanobacteria also. But as it is a algae, so it's a blue-green algae. The pigments are blue and green. Okay. What is blue? Phycocyanin pigment. And chlorophyll is a green pigment. And algae are of different types. It's not compulsory for every algae to be a blue-green algae. They all are having different structures like Cara. I remember one algae, Cara. Cara is a green algae. Okay. There are some red algae also. They are having red pigment.
Some brown algae is also.
Brown algae is also there. Rhodophyceae is the pigment. Basically, I'm not a botany teacher, but I remember all these things.
Yes.
I studied in the my class, my student life, in my student age. Okay.
Yes.
Any other question? Algae are basically characterized according to the structure and their pigments. Every algae is having a different kind of structure, reproduction, and everything. Okay, let's ask some question to you. Have you seen a silverfish? Has anyone seen silverfish? Silverfish.
No, ma'am.
Do you know silverfish is an arthropod? Silverfish
Yes, it
is
is
a white-like organism. Whenever you will take out a book after so many years, some book is lying in your shelf, either it is of your parents, and you take out a book and you will find a white-like organism is going out of the book. That is silverfish. That is arthropod. Okay? It is not a fish. Fish comes under the chordate class Pisces. Okay? It's a arthropod. This is a general question. It just came into my mind and I just thought I should discuss this with you. Silverfish is a arthropod. And prawn is a fish. Prawn, jhinga. Normally called as jhinga machli.
Aquatic animal.
Aquatic animal.
Arthropod. Aquatic animal, but
Yeah,
it is an arthropod. It is also not a fish.
Yes, arthropod.
Arthropod. You will be doing
Yes.
dissection. You will be doing dissection of prawn in your graduation. Okay. In the first year of your graduation,
What
you
will we
will
do,
do
ma'am?
dissection of prawn. Yes, tell me.
What will we do?
What? Beta, you are not clear. Your voice is not audible.
What will we do with prawn?
What we do with prawn? Prawn is the favorite seafood of many people. Those who love seafood. Those who love seafood, prawn is a seafood.
In
It is
graduation, what we will do, madam?
what?
In graduation first year,
Yeah,
what we
ma'am.
will do by the
My
prawn?
question.
You do the dissection of prawn. You took out the endoskeleton of that. Arthropods are having endoskeleton. They are having their body is divided into three parts.
For
Cephalopod,
frog also.
dissection, your teacher will tell you, do the dissection, take out the alimentary canal of prawn, take out the excretory system of prawn, and you have to do. Your teacher will explain you how to do it, and then everyone will have to do. Teacher will come to your seat and she will check you have done it right or not. This is the practical. What you do, there you don't have to eat, because all the specimens, all the organisms that are used for the dissections are kept in the formalin, and you can't eat that formalin one. Okay, formalin is a solution in which your all the insects, dead animals that are used in the biology lab for dissections are kept in formalin. There you are not about to eat it. Okay, but prawn is basically favorite food of many people, like crab. Many people eat crab too. Like I am a pure vegetarian person, but I know this is a sweet food. Okay. Is there anything you want to ask?
No, ma'am.
Okay, then let's end the class.
Yes, ma'am.
Okay, you continue with other class. If there is some other class, you can go with it.
No, ma'am, we have only one session. Evening time.
Okay, fine. Then study hard, do the hard work, and don't study too much, but study the important things. Make short notes, make bullet points. Every and
Yes,
use as much examples you can take out from anywhere. Okay. Always learn the diagrams, because diagrams help you in understanding the things more easily. Bio is a subject, their diagram is very important. Okay. So you need to practice a lot. And when it comes to the phylum needs lots of hard work to memorize the every characters of phylum. Okay. That you have to do. Biology needs labor. You have to learn to memorize the things.
yes.
Okay. So all the best to everyone. Prepare this topic. I'll be asking some questions tomorrow. Okay. And
Thank you, ma'am.
if any query, then keep that query. I will clear that query tomorrow. Okay.
Okay.
Okay. Bye-bye.
Bye, ma'am.
Yes, ma'am. Thank you, ma'am. Yes, students. Thank you, students. Again, we will meet tomorrow morning session. That is 5 to 7. And students, please revise whatever madam and sir are discussing in the class. Please don't forget. You are getting this time 8 to 9. You are free, so please revise in this time.
Okay, ma'am.
Yeah. And please join the morning sessions 5 to 7. Those are also very important. Be serious. Okay. Bye, everyone.
Bye, ma'am.